All right, we're going to give it a moment to make sure that everyone is able to get into the webinar. I see colleagues from Georgetown here. Great to see you. We're going to wait a few more moments and make sure that everyone's able to get in. Thank you for your patience. You can see our distinguished lecturer is already here, Dr. Landau. Hope you all are feeling well on this beautiful sunny day here in BC. This morning it was a little overcast, but it looks better outside now. It's great to have so many people joining us today. It's great to see all of you here. Get started with some, what's the phrase? Some housekeeping is the phrase in English. And then we will have a formal introduction and then have our distinguished lecturer begin her talk. So if everyone could hold their questions until the end of the presentation, that would be great. You can ask questions then. You can submit questions at any time though through two ways. You can use the Q&A feature, which you should see on the bottom of the screen. You can type in your question there. And then if you want to sign your question in ASL, you can enter in that you want to do that and we can call you on screen. Or you can type your question in English and we can read that for you. We also do have Asl English Interpreters here. If you require interpretation, if you are coming on screen and you want to use spoken English, let us know just to make sure we have the interpreter on screen. We do have captions. I will share the link for the captions now. I think you have to open up a separate screen to see the captions. My understanding is that's a lot faster and generally better quality than if we use the captioning feature innate to Zoom. I believe that both will work though. I would now like to ask Lauren to turn her video on so that we can have our formal introduction of our lecturer today.
Good afternoon. I am delighted to have the honor and privilege to present today Dr. Barbara Landau, who has been selected to be a distinguished lecturer for Gallaudet Universities and the PhD in Educational Neuroscience Penn Program's Distinguished Lecture Series in Educational Neuroscience. This Distinguished Lecture Series is an important annual event to recognize and honor truly world-class leaders in the cognitive and educational neurosciences. Our theme this year is from brain development to developing virtual brains. Today, we are especially thrilled to have Dr. Landau accept our invitation as our distinguished lecturer Dr. Landau is a Johns Hopkins Gilman Scholar, Distinguished Faculty, and the Dick and Lydia Todd Professor in the Department of Cognitive Science at Johns Hopkins University. She is also an elected member of the National Academy of Sciences, a Fellow of the American Academy of Arts and Sciences, the American Association for the Advancement of Sciences, and the Cognitive Society, and was named a Guggenheim Fellow in 2009. From 2013 to 2018, she was the director of the Science of Learning Institute at Johns Hopkins University. Dr. Landau received her Bachelor of Arts degree in sociology from the University of Pennsylvania, her Master of Education degree in educational psychology from Rutgers University, and her PhD in psychology from the University of Pennsylvania. Dr. Landau is known for her research on unusual cases of development, such as congenital blindness, Williams syndrome, and perinatal stroke. She is a leading authority on language and spatial information in people with Williams syndrome. Dr. Landau is interested in human knowledge of language and space, the relationship between these systems, and how they are represented in the mind and brain. Dr. Landau's distinguished lecture title is Genes, Brains, and Spatial Representation, Evidence from Williams Syndrome. Please help me welcome Dr. Landau as our 2021 Distinguished Lecturer. It is indeed our great honor to have you here. Welcome to Gallaudet.
So shall I go ahead and share my screen now?
Yes, please do share your screen.
Okay. Thank you so much for that lovely introduction. I am really delighted and so honored to have been invited to give this talk. And I hope that that you'll enjoy it, that you'll find it interesting, engaged in for, it seems like almost 2 decades now. Hard to, although we won't be having questions interspersed with the talk, I look forward to your comments and questions at the end of the talk. So I'm going to start out with this title page, and I'm going to point out to you that this profile that I've shown here is very characteristic of what I would call the huge difference in capabilities for people with Williams syndrome between their visual spatial abilities and their linguistic abilities. And the graphic just shows that if these are actually data from our lab. If I show a person with Williams syndrome, an adolescent with Williams syndrome, this figure, and I ask them to draw it, I might get something like this, where it's clearly got the right colors and it's got the piece bifurcated in the middle, but the organization of the spatial properties is really distorted. If I ask the person, what do you see here? What is it? They might say it's a circle with the blue above the red. So they are very, very capable linguistically. And I'll be talking about that profile throughout, asking how do we get there from knowing that that Williams syndrome is a genetic condition. So I want to start with the profile as presented to us in the 90s by Ursula Bellugi. She's really the person that brought cognitive scientists brought Williams syndrome to the attention of cognitive scientists. And what she showed was something that was similar to what I just showed you, that it looks like space and language are completely separate systems. One is really targeted by the genes and the other one is not. And this was the way that Bellugi interpreted the data. This is another example of a big distortion in when an individual who's 18 years old is asked to draw an elephant, clearly there's not very much at all of spatial organization. But if you ask them what it is, they say what an elephant is, it's one of the animals, it lives in the jungle, it can live in the zoo, it has long gray ears, fan ears, they can blow in the wind, and continues in a very, very articulate fashion. So Bellucci actually proposed this to be a very interesting case where one could view the case as producing a cognitive profile where one system, the spatial cognition system, is really targeted, is very, very deficient, whereas language is completely spared. And this would indicate to her at the time that they were evidence for modular systems that were differentially targeted by genes. You know, I'm sorry. I'm going to give you, I'm having some problems with advancing the slides between my cursor and my mouse. So just be patient, please. This is an example. It's a short video of an adult who is actually doing a task, a block construction task, which is part of many standardized IQ tests. And the reason that I like to show it is to give you a really sort of clear sense of what this deficit really looks like. So in this case, there's a flat design here. It's actually made up of, you can see there are two black triangles, and then there are a number of areas that are square yellow areas. And the person is charged with taking these blocks, which have different designs on each side, and forming the blocks together so it makes that pattern. So I'm going to play the video for you. There's a little bit of audio too that you might have difficulty hearing, even if you are capable of hearing. And so I'm going to just over talk what is going on, and it can be signed. Hold on, just, oh, sorry, this is giving me a problem. Okay, here we go. So the experimenter says, try it, see how you can do it. Use the blocks to make the design. And you can see that the individual who is doing it, who's a young woman who's in her 20s, is going to look at the blocks and look at the design, and now she's going to start. And what you'll see is that she puts together two blocks that actually have the right patterns on them. So she's going to find on those 6 sides, she'll find the diagonally split blocks with half green, half yellow and half black. Then she'll pick up two of the solid blocks, which is also correct, and she'll organize them, but she comes up with overall the wrong pattern. And the experimenter then asks her, what do you think? How is that? And she says, very good. And she's asked if she wants to try again. And the answer is no. People always want to know, do the individuals know that they really have problems, that they struggle with these kinds of tasks? And the answer is yes. When we give these block construction tasks to people with Williams syndrome, they typically say, oh, I'm terrible. In fact, they do struggle with doing these problems. So that's very characteristic of the spatial impairment. So just to tell you a little bit about background for Williams syndrome, it's a genetic deficit. You probably already know that. It's a microdeletion on the long arm of chromosome 7. It's about 28 genes and it contiguously deleted. And there are a variety of phenotypic characteristics, mild to moderate retardation. But for our purposes, of course, we're interested in characteristics of brain and cognition. Severe impairment in visual spatial construction, but very good language, as I said. Overall, there's brain volume reduced. That's similar to many cases of people who have intellectual challenges, difficulties, deficits. For Williams syndrome, the occipital posterior parietal areas, the areas that actually are very important for spatial organization, are relatively small. There are some structural abnormalities. And there's also abnormalities, both functional and structural, in the hippocampus. And all of this, the dorsal parietal area areas and the hippocampus put together indicate that clearly the brain is representing, is causing or some kind of a spatial deficit. The genes deleted in Williams syndrome luckily have been identified. It's been almost completely mapped in this contiguous area. And also, fortunately, the mouse chromosome 5 represents exactly those same genes. And so this has, in a contiguous area. So this has actually enabled geneticists and behavioral scientists to try to associate different individual genes with aspects of the behavioral deficits. There's been limited progress on that. I'll just say that there are a couple of genes that I've put an asterisk here for these two genes are associated with learning and memory dysfunction. And then there's another gene that has been indicated as possibly involved in high sociability. It's been experiments in dogs who are missing that particular gene. So there's some progress, but it's not enough for us to really have the goods on what the genes are doing and why they're causing this very unusual cognitive profile. So the big question that I want to ask today is, how could missing genes affect cognitive structures? And I'm going to start with this. We've got some missing genes. I just listed them for you in the last slide. That leads to formation of a brain. It's going to be a different kind of a brain in some ways than an individual without the missing genes. That leads to cognitive structures, and the cognitive structures then lead to behavior. And the behavior is just the behavior that I showed you on the very first slide, that is to say, an impaired spatial ability with a very, very high functioning language system. And so my question has always been, what can we say about cognitive structure? So the key here to me is to try to understand cognitive structure and then to work our way back to what the brain might be doing. And at the same time, to interpret cognitive structure in terms of the actual behavior that we see. So if I come up with an explanation at the end of this talk, I'm going to owe you an explanation in terms of what the behavior is that's actually produced by these individuals. Okay, the plan of the talk is this. And again, I'm apologizing because my mouse and my cursor are not behaving themselves. So we're sometimes having to backtrack with slides. The plan of the talk is this. The first thing I'm going to talk about briefly is the causal chain. What do we think about the possible causes here? What are the puzzles we're trying to solve? And I'm going to offer two hypotheses. The second thing I'm going to do is give you evidence on the spatial cognitive profile in some detail for people with Williams syndrome that we've accrued over the past 20 years or so. I'm then going to offer you a speculative hypothesis focusing on typical developmental trajectories. That is not the atypical ones of the Williams syndrome, but typical developmental trajectories. And those typical developmental trajectories, I'm going to say, are part of the explanation for the profile of Williams syndrome. And that position makes some surprising predictions for language and other domains that I'll lay out for you. And finally, I'm going to revisit this gene cognition causal chain. So starting with the causal chain, the puzzle is, how do we account for the striking apparent dissociation in Williams syndrome between language and spatial representations? They look very, very different. And how do we talk about the role of genes? How can we account for this in terms of the gene cognition causal chain? So there are two hypotheses I want to put on the table. One is what I'll call the modular hypothesis. And this is the idea first articulated by Ursula Bellucci that genes target particular cognitive systems. In this case, they would affect spatial representation as a set of systems, and they would not affect language. And that modular hypothesis was offered first by Bellucci. It's also been endorsed by linguists such as Ray Jackanoff, Steve Pinker, and Harold Clausen. And the focus here is on domain specificity, that the particular things that the Williams syndrome individual can or cannot do, struggles with or does not struggle with, are criteria for modularity. That if you can knock one out and preserve the other, it must be that the two systems are quite different and possibly modular. The second hypothesis, which I'm going to spell out today, and actually I'm going to end up endorsing, is our hypothesis, which I call the developmental hypothesis. And that is that the genetic deficit does affect early mechanisms of development, for sure. There's a change in the brain. That in turn, overall affects the timing of emergence of cognitive systems. And I'm going to use that developmental hypothesis after I put the data on the table for you to try to explain how it is that we got this very unusual profile of an 18-year-old who simply can't do a 4-year-old a simple block puzzle, but can nevertheless give you very, very fluent and articulate language describing that puzzle, for example. So the approach.
Sorry,
That's okay.
I've received some requests to tiny bit slow down because
Sure.
It's a little fast, just a tiny bit. Thank you. Sorry so much.
Sure. And let me know again if I haven't slowed down enough, please.
No worries. Thank you.
So the approach that we've been using, that my lab has been using for many, many years now, it is embodied in these 3 points. First of all, we think that you have to look broadly. That is to say, to understand why space is impaired, you can't just look at space. You have to look at it within the same individual relative to other systems. So I'm going to look at space and try to understand space, but I'm also going to try to understand language. Secondly, we want to look in detail. So we take every single task that we use and we examine its microstructure. We examine it very, very carefully. We don't do things like simply using overall intelligence tests or overall vocabulary tests. But for every task that we have, we try to think in detail, how is this task being performed and is it different? Is the person performing it differently from typically developing individuals? And the third principle that we use is to look developmentally. And by that, I mean that we pay attention to the timing of development. And we ask, can the differences be explained by differences in the timing of typical developmental trajectories? So I'll return to these things. You don't need to remember them all in detail at this point. But that's the approach that we've been using. Okay, in terms of data that we're going to be looking at, we're going to have 3 possibilities that we're going to consider in terms of the outcomes. One is the idea that there is what I call persistent structure, that the individual with Williams syndrome may perform at a lower level in terms of age equivalence, let's say, but actually the performance is qualitatively the same as a younger child. The deficits are only quantitative, not qualitative. And that emphasis is on the idea that emphasizes the idea that there may be constraints internal to any cognitive system that have the outcomes be qualitatively the same, even though you could do better or a little bit worse on a particular task. Another possibility that we can consider is that there's just absent structure. And this would be the idea that if there's a really terribly impactful spatial deficit, then perhaps we won't find anything that looks like normal structure in the behaviors of the individuals. And this emphasis would be on the tight link between genes and cognitive systems, as if the genes that are missing really have knocked out that knowledge or that cognitive structure. And finally, the third possibility is that there's qualitative change. And this is something that many, many groups and people who study intellectual and cognitive impairments subscribe to, the idea that there's really a different system going on. It's not the same as a typically developing individual. It's really a different cognitive system. And this would be the idea that when the genes take their effects, there are cascading effects such that you can never get the same organization, the same structural organization of cognition, but rather it will be something that is just not seen in any other circumstance. So I'll tell you in advance that the 2 that we're going to have evidence for is persistent structure on the left and very little evidence for qualitative change, probably no evidence, and some question marks about absent structure. We think that it doesn't exist, but there may be some question marks there. So mostly I'm going to be emphasizing persistent structure, as you'll see. Okay. So I'm going to talk about 4 different cases of experiments, and I'm actually going to walk through these experiments relatively quickly to give you the bottom line on what the phenomenon is. One is object recognition. Can we recognize objects from different perspectives, and how good is that? The second is on biological motion perception, which many of you, I'm sure, know about, but for those who might not, this is the human being's remarkable ability to take motion trajectories from simple individual dots, and when they undergo motion, to give you immediately a perception of, for example, in this case, a human doing a backflip. So it's biological motion. The third thing I'm going to talk about is vision for action. This is simple actions in which, in this case, the child is going to be sticking a rigid dollar bill through a slot. And the fourth is reorientation, which has been a highlight of a great deal of developmental work in the past 15, 20 years, where one can or cannot recover their orientation after having been disoriented. And I'm going to talk about those 4 cases to lay out the data, the facts of what the Williams syndrome spatial profile looks like. So here I go. Case 1 is object recognition. And again, the idea here is, the question is, do we have, for those of us who are typically developing or developed adults, we can recognize a carrot from its so-called canonical view. And we can also recognize it from unusual views. In both cases, it's a carrot, and we can name it a carrot. It may be harder from the unusual view than the canonical view, but you have to have equivalence in order to have full capacity object recognition as an adult. This capacity engages multiple viewpoint representations. That is to say, you have to be able to represent or store what an object looks like from different perspectives. It's known to be housed in the inferotemporal cortex. And the unusual views, when one is recognizing unusual views, that very often engages parietal areas as well. And that's the area, of course, one of the areas that seems to be structurally impaired in people with Williams syndrome. So let me show you the experiment. I'm going to show you some clips, and you can just guess to yourself what the object is. I'm going to say them out loud as they come, but this is the way the objects are presented. Half a second exposure for individual objects that either appear in canonical or an unusual view. And you just have to name it. That's the task. And we've done this with 4-year-olds, typically developing 4-year-olds through adults, and also with people with Williams syndrome. So here goes. Violin, anchor, hanger, sunflower, dresser, trumpet, pumpkin. Okay, so those are some examples of the objects that the children and individuals with Williams syndrome see. This study was done with, I'm sorry, I have to remove this bar from the bottom of my screen. Okay, so the individuals with Williams syndrome are on average 11 years old, and the mental age matches. These are children who have the same mental age, or about 6. And we have here plotted the percent correct. First, look on the left to the full color clear images. We have the canonical orientations, and we have the unusual orientations. This is a stool from underneath. This is a stool just looking upright. And what you can see is that the canonical orientations for both chronological age matches to the Williams syndrome individuals, that is kids who are typically developing kids who are 12, are way up at the top, are performing basically at ceiling levels, as are normal adults. And you can see that the Williams syndrome individuals and the mental age matches also are doing quite well. It's all above 90%. So these recognition of canonical oriented objects is totally at ceiling by the time the Williams syndrome individuals are 11. On the other hand, you can look on the other side for the unusual perspectives, and you see a huge difference. So first of all, everybody decreases in their performance, all groups. But what you see with adults and typically developing 11-year-olds is they're still 90% correct and above. They're still doing very, very well. But Williams syndrome individuals who are also 11 or 12 and mental age matches who are 6 really plummet. So they're down now around 75%. So these are much, much harder for these individuals who are 11 years old, the Williams syndrome individuals who are 11 years old, and about the same as the mental age matches. You can look at the right-hand panel if you want, but it's not crucial. That just says the pattern replicates perfectly if you do line drawing, so they're not capable of using things like color to identify the objects. So what that tells us is that canonical orientations are really easy, and at ceiling levels from the time you're a 6-year-old, actually from the time you're a 4-year-old in our data, all the way up through adulthood. But unusual views really develop. It changes over time. So the sum is, just from that one picture of data that you have, the canonical view, as I said, typical development actually asymptotes by about age 4. It's an early emerging function of the visual system. The unusual views system is really much more late emerging. Typical development asymptotes way later, probably after age 12. And what you see is that the Williams syndrome children and adolescents, that is Williams syndrome individuals who are around 12 years old and span between 10 and 17, they identify both canonical and unusual viewpoints at the level of a typically developing 5 or 6-year-old. Okay, so it's not that they can't do it, they absolutely can do it, but they are showing a pattern that reflects the same as a typically developing child, much, much younger. So for that piece of data, that set of data, we say that's persistent structure. That reflects persistent structure. So the deficits are quantitative, but they reflect typical cognitive structures. Again, the way the visual system works, it works the same in people with Williams syndrome as in typically developing, but there's a quantitative difference in how well you can do at different ages. The second case is biological motion perception. And as I said, this is this remarkable ability to just take a set of dots, and once they start moving, it emerges as a moving biological entity. And we know that babies are sensitive to biological motion, and also that it can be selectively damaged when somebody has brain damage as an adult. There are specialized cortical computational mechanisms that do the job of biological motion. And it probably has a special house in the brain, in the superior temporal sulcus area of the brain. So this work, we went to look at whether or not people with Williams syndrome had any kind of ability to do biological motion perception, and if so, whether it looked like the typical case or completely different. So the design of our study in biological motion was this. We had 2 different kinds of signal to noise ratios, and we had 3 different kinds of noise. So this is an example of a signal to noise ratio of 1:1. There are the 11 dots that form the human figure, and then there are 11 dots that are noise dots. The static noise is just, as you'll see, static noise. It's just the dots are there, but you can look at it and you can guess which way is the point light walker walking. And probably everyone can see pretty clearly that the walker is walking to my left. It should be to the viewer's left. Okay, pretty easy. We'll take you now to the next most difficult condition, which is again a 1:1 signal to noise ratio, the 11 dots with another 11 noise dots, but this time the noise is random. So although the point light walker still has its internal motions, the noise dots are going in random directions, and you'll see how you can do with this one. It might get a little bit more difficult. Which way is the point light walker walking? And in my view, from this angle anyway, it looks like it's walking to the right, but it should be relatively easy, but a little bit more difficult than the first because of the noise type. And finally, I hope I'll give you the next one. This cursor is really misbehaving today. Okay, the 1:3 signal to noise ratio with yoked noise. That's the hardest condition we have. For every 1 dot of the point light walker, 11 dots, there are 3 different noise dots that go with that dot. And the noise of the noise dots are yoked to the same trajectory as the signal dots. So you'll see it's going to be very hard to extract the figure and its direction. And here we go. And depending on your viewing distance and your expertise and this kind of thing, you can get much better at it. But for most of you, it should be very, very hard to do. And some people might be able to do it. So we've got different levels of difficulty where we're going to compare typically developing adults, people with Williams syndrome, this time around 12 years old, ranging from 9 to 15, and mental age matches who are on average 6 years old. This is with the 1-to-1 signal to noise ratio. And what you can see is the adults, typically developing adults, 18-year-olds, and Williams syndrome individuals 9 to 15 are basically indistinguishable. They're no different. The mental age matches, who are 6 years on average, are very good in the static case, the case of static noise, but it's harder for them to do random or yoke noise. And that's in the 1-to-1 signal to noise ratio case. When we go over to the 1-to-3 signal to noise ratio, which is much more complex, we see actually very similar patterns. Everybody gets worse as with increasing noise, with different harder kind of noise. So every group shows this decrement, but what you can see again is that the individuals with Williams syndrome are very, very close to the adults. There's no difference for the static noise. There is a significant difference for the random and the yoke noise, but again, the Williams syndrome individuals are more similar to the adults than they are to the mental age matches. So this is a very unusual case where individuals with Williams syndrome who have a severe spatial deficit turn out to have a profile for biological motion, which is more like fully developed adult controls than it is like their mental age matches. So something there is really preserved in their ability to detect those, the point light walkers. So this is another case of persistent structure, as I will argue, that the deficits again reflect typical structures. They have to, the computational system in their brain must be capable of extracting that form and detecting its direction of walking. And that's a specialized mechanism. They can't do it using any fancy strategies. And so that suggests to us that there are very, very strong constraints on that knowledge system, and it's preserved for people with Williams syndrome. The third case is going to be vision for action. And in here, we actually looked at both vision for action and vision for perception. What we did was we took a setup where we had an oriented slot you can see in a board. And this here it's horizontal, but we also had vertical and then some obliques. And in the case of vision for perception, we asked children, we attached the dollar bill to a little mannequin hand, and we dialed the orientation that the hand was holding the bill. And we just asked the child to tell us when the dollar bill was ready to post, and they would say stop. And that was perception, matching the perception of the dollar bill to the slot. In the other case, we just asked them something that for any developmentalist, you'll know it's totally simple. You just ask the child who's as young as 4 years of age to pick up the dollar bill from the table and to put it in the bank, okay? And we measure the angle at which it hits the slot to determine how accurate the child is. And what we find in this set of studies is that in typically developing individuals, 4-year-olds actually are at asymptote. They're very, very good for perception. But only 6-year-olds approach asymptote for action. It's much harder for them to get it into the slot appropriately than to judge when the orientation is correct. We also found that Williams syndrome adolescents and adults are more like 4-year-olds, typically developing 4-year-olds for action and perception. And I want to give you some examples of that, of the action data in just the next slide. The first thing I want to show, I'm sorry, this cursor is really going wild. It hasn't happened before. It might be it's the space heater that's on here. In any case, the first thing to look at is this is vision for action. This is the angle at which the dollar bill goes into the slot. So for degree 0, the vertical, a typically developing 6-year-old child pretty much gets it right on the mark. Same thing for the horizontal slot. They orient it properly. And then the 2 obliques are pretty good too. They're pretty well tuned to their slot. The slot is underneath it. So there's more noise here in these 2 cases than there is here or here, but they're still pretty good, typically developing 6-year-olds. Children with Williams syndrome on mean age of 12 show much, much noisier data. So at degree 0, they're well tuned to the slot, but they're not perfectly tuned to the slot. You can even see that some of them are going for a horizontal orientation. For the horizontal, again, they're tuned to the slot, most of them, but there's a huge amount of noise. And then here, for 135 and 45 degrees, the 2 obliques, they're very, very noisy. So again, they're tuned to the slot, but there's a lot of noise. It was at this point in our working on Williams syndrome that we started to ask the question, well, do these children ever outgrow this property? Do they get better when they get older? And so what we did was we also tested adults with Williams syndrome. And we found this pattern, if my cursor will allow it. Wow, okay. This is with Williams syndrome adults, almost 24 years of age. These are the different slots that they're trying to get the dollar bill into. And you can see it doesn't look too much different from the individuals who are 12 years old. 24-year-olds are still having tremendous difficulty getting that dollar bill into the slot. They're still looking much noisier than a typically developing 6-year-old. And what we then went to ask is if they don't seem to persist and get better in this task, what is the equivalent here? What is the equivalent for Williams syndrome children who are 12 years old? And so we said, they're worse than 6-year-olds. Let's go down in typical age and see what happens. And so we tested 4-year-olds, 3 to 4-year-olds. And if you can look down here in the lower left panel, you can see that for the different oriented slots, they're tuned to the slots the way the adults are, but in fact, they show as much noise as the Williams syndrome adults and the Williams syndrome children. So putting it another way, typically developing 4-year-olds, 3 and 4-year-olds are roughly the equivalent. That's where a Williams syndrome individual gets to in this particular task, this spatial task. If you're 12, you look like a 3 to 4-year-old. And even if you're 24 in this task, you look like a 3 to 4-year-old. But I want to point out that even though you look quantitatively worse, for sure, because there's more noise, the tuning to the actual slot is still there. You still have the ability to roughly tune, but there's a lot more noise. Okay. So this case, again, we're going to say reflects typical structures. There's persistent structure there. It might look a lot worse than an average, typically developing 12, 18, or 24-year-old. They are much worse than even their chronological age matches who are adolescents at 12. But they're very similar to typically developing children who are much, much younger. So that's going to be persistent structure again. So those 3 cases so far are all persistent structure. Now let's turn to geometric orientation. I want to be mindful of time because I am going a bit slower and I may end up skipping a little bit of the material so we can get to the end. This is a different kind of task. This is an orientation task, a reorientation task, where after being disoriented in space, and by that I mean cover somebody's eyes and turn them around in space, you will find if you do that, you're very disoriented. You don't know where the door is, for example. When that happens, whether it's to toddlers, adults, or the many, many non-human species that have been tested, what's been documented, well documented, is that all species reorient themselves using the geometric properties of the layouts. That is to say, the shape of the space, not the specific surface properties. So this was first discovered with rats and by Cheng and Gallistel. And they found that, in fact, the rats produced this unusual pattern where if the goal was hidden right here, then the rat would search at the correct location, but also at the geometrically equivalent location, which has the same configuration of a long wall to the left, a short wall to the right. Here again, it's a long wall to the left, a short wall to the right. So these 2 corners are geometrically equivalent in the array. So they're geometrically equivalent, but even though they have very, very different surface properties, so for example, this could be a lit panel, and this could be an odor, and this could be a different a set of geometric figures, and this could be another one. They could all be very visible and very salient to the animal, and yet they ignore those properties, the surface properties, and just use the orientation just do reorientation on the basis of the rectangular shape of the array. So we know from a lot of developmental work that use of geometric properties appears in babies by 18 months of age. What's longer lived is the combination of that geometry, later developing is not the use of the geometry, but the combination of geometry with the surface properties. So for example, individuals who are typically developing 18-month-olds will use the geometry, but not the landmarks. Whereas 5-year-olds seem to be able to use both of those things. And then they give you a pattern where they don't show you this very characteristic geometric equivalent search, but rather they'll look in the right spot because they're combining both the geometry and the salient surface properties. So our findings with Williams syndrome individuals show that children and adults both can do geometric reorientation. And they can do that, but they have great limits in combining the geometric representation with landmarks per se. So I'm going to show you a few experiments that demonstrate that. I'm going to pass this slide just quickly, just to say there was good reason to do these studies in people with Williams syndrome because of some studies with knockout mice that showed that there were orientation problems in mice who were missing the particular gene that was suspected to be associated with spatial orientation. And also, we thought there was good reason to do the experiments because of these abnormalities in the hippocampus and the parietal that had been discovered in people with Williams syndrome. So the experiment goes like this. There are 19 people with Williams syndrome. Here there are two different arrays. One is an all black room, no landmarks at all. So there you have, if you're going to be disoriented, you're probably going to be choosing between the correct and the geometric location. The other room actually has a landmark. So you can use geometry and landmarks and you can get it right. So we do it with people with Williams syndrome, age 17 years on average, and 12 typical. We have the object in the corner. We then disorient the participant by blindfolding them, turning them around in circles, stopping them at one of the walls, and then allowing them to go and find the toy. And this is what happens. In the all black room, where there are no landmarks at all, typical adults use geometry. So you can see that their responses are split between the correct corner and its geometrical equivalent. People with Williams syndrome do not do that in this experiment. So you can see that these searches are randomly distributed. There's no very salient correct plus geometric responses. So it looks like they don't use geometry. This was our first study. We also looked at the case with one blue wall. And in this case, typical adults do perfectly because they can combine the geometry with the surface properties, the blue wall. It looks like the same individuals with Williams syndrome can also do it, not as well as typically developed adults, but pretty well. And it suggests that they are able to use the landmark, but they weren't able to use the geometry. So they can't be doing some kind of combination in the same way that a typical healthy adult would do it. That was where we stood with the data until Katrina Ferrara came along, and we worked on using slight modifications of the task to see whether or not we could really push these individuals to see, could they detect the landmark, could they detect the geometry, and could they combine? And she did very, very small changes to the arrays. In particular, she did things like, instead of hiding the things behind a panel, she hid them in little canisters. She strung little Christmas lights around the top so that the whole arena was a little bit lighter and brighter for the children. And we now tested a very wide variety of aged kids with Williams syndrome between 5 and 32, very large span, altogether 16 participants. And we also tested very young, typically developing children between 3 and 4 years old, who should in principle be using the geometry and maybe sometimes also combining with the landmark. So let's see what happened. This is insisting on jumping ahead. Okay, the room with all black walls. With typically developing children definitely show this characteristic correct plus geometric or rotational equivalent. So they're showing the standard case, the standard pattern of choosing on the diagonal here at these two geometrically equivalent corners and doing that well. And it turns out in this revised experiment, so did individuals with Williams syndrome. They did really quite well. So they showed that they could use geometry as well in this new format that Katrina used. So good, we've solved that problem. It looks like you have to make things sort of optimal for them to be able to use the geometry, but use the geometry they can. Then we asked about the feature. What about the room with the one red wall? So here, remember that adults will absolutely get it correct because they have both the geometry and the feature or the landmark, probably the feature. Typically developing 3-year-olds, some of them can. So there's not an exact equivalent between the two corners. They're actually doing better. But look at Williams syndrome. They're really doing much better. They really can seem to use the landmark as well as the geometry. We just saw that they used the geometry. Now it looks like they can combine it with the landmark. But we were still very curious because our previous work that I told you about showed, one, it was hard to use the geometry. You had to make things optimal. And two, it didn't really look like they were able to robustly combine geometry with landmarks. So what we did was we did an analysis, which we called the beeline analysis. And so what we did was we tried to see how efficient are they in finding the target in the all black or the red wall rooms, in the two different rooms. And this is compared to your Williams syndrome and typically developing 3 to 4-year-olds. And what we did was we thought to ourselves, well, if the target was placed in this corner right over here, upper right, and if you ended up and had your blindfold removed at this wall, facing this wall, well, then you might have just to turn one wall. And that would be the most efficient way to detect that it was in this corner, to compare it to the right representation and decide that was the place. But you also, if you turned in the other direction, you might have to go to this wall and then this wall and then this wall and back to this wall. So really we were testing, are people taking that super efficient route, which you would use. You would take the super efficient route if you really had a representation of the whole thing and had just a bit of information and were immediately able to go to the correct canister. And what we found is this is an average difference score between the optimal path, the shortest path, and the path they actually took. And so scores around 0 or 1 suggest that it's a very efficient searcher. They're basically just doing either just one wall or even 0 walls if they happen to open their eyes at the right wall. And this is plotted against the proportion of searches to the correct or the rotation. So it's plotted against how well they're doing. And what you can see in this case, this is the all black room. And you can see that most people, including Williams syndrome, individuals with Williams syndrome, have a pretty robust, efficient beeline responding. Using just geometry, they're really efficient. They go immediately to the first corner or the rotational equivalent. With the red wall in combination, in the case of the red wall room, they actually don't do that. So what you see is that typically developing children are still using the beeline process. They're only searching using one wall. They're traversing one wall to get to the right corner. But the people with Williams syndrome are showing that they're going around and around. They're searching, and so they might start facing this wall, and then they might look to this wall and this wall and this wall and this wall and this wall, and end up with a score that's around 6. So in other words, 6 different walls that they've searched before they've settled on their solution. So what that says is people with Williams syndrome are basically not combining things in an immediate fashion. They're not combining geometry and landmarks immediately the way typically developing 3 to 4-year-olds do, and the way typical adults do. We ran typical adults, 18 and older in this, and they're at beeline for both cases. They're just immediately, they have a representation of the array and they immediately turn to it. Okay, so here we're going to go not only persistent structure because we have the geometry, they're capable of geometry, but we're also going to raise the question mark of, is there an abnormality here? Is there a problem? And do we have a different kind of system? I'm afraid that I'm actually going to skip some of this. I might give you just one little snippet about the boundary work, but we do have evidence that people with Williams syndrome also have trouble with just defining boundaries. So just to give you one simple little snippet, as another piece of evidence that they do seem to have some problems with sensitivity to boundary structure, we know from typically developing kids that when they're in the geometry, in the reorientation task, if there's a very tiny curve that constitutes the boundaries, we've always watched them in walled rooms and they're good at using geometry, but they can even do it at 4 years old with just a tiny little curve. So they're very sensitive to the idea that there are boundaries, and they're still doing this geometric search pattern. With a mat, with a solid thing on the floor, they absolutely don't. These are just typically developing 4-year-olds. It turns out that when we test individuals with Williams syndrome, they do not show sensitivity to the curve the way typically developing 4-year-olds do. So they're good when you have full walls. They can use geometry when you have full walls, but they can't do it with a curve the way a typically developing 4-year-old is capable of doing. And I'll apologize in advance for skipping these data, but I think it's better to just move on to the conclusions that we have from this and sort of explanations. So let me just say the summary for geometric or reorientation is, again, there is persistent structure. They can do geometry, but there might be some qualitative changes. There may be differences where combining the two pieces of information is really much, much more difficult for them. Okay, so back to the original question. How do missing genes affect cognitive structures? Missing genes, we've got a different kind of a brain, we've got cognitive structures. What are those structures like to produce this particular very unusual behavior? So a review of all the effects, not all of the effects. Some of the effects I've told you about. This list is actually a list of all the effects that we've found. So we talked about object recognition and biological motion. And we found that there's persistent structure for sure for these two systems. We also know that individuals with Williams syndrome have really strong language. They have good face perception. They actually are very good at what we call intuitive psychology. This is tested by asking them questions about whether a child is actually playing with another person, engaging with another person, even though you can't see the other person. In all of these tasks that we've done with them, we see what we call persistent structure. Their age equivalent is they're as good as a typically developing person who is a mental age match, or even better. For the quantitative change, there's a whole host of things. So we looked at vision for action, where people were more like typically developing 3 or 4-year-olds. We've done multiple object tracking, where you have to track a number of objects as they're all moving together and identify which are targets. They have a capacity to track up to 3 objects, whereas their age equivalents are able to do 4, 5, 6, or 7. We've worked on subitizing, the ability to immediately abstract the number of a small set of objects. They have a subitizing range that is similar to 3 or 4-year-olds. We've done work in the approximate number system, where we've found that their ability, their sensitivity to doing estimations of number is very much in line with 5 to 6-year-olds typically developing. These are even among adults with Williams syndrome. Intuitive physics also is at the level of 4 to 6-year-olds. Praxis and copying, I showed you some data before, but even in standardized tests, their age equivalents are sort of 4 to 6. And then I have as a question mark whether boundary sensitivity and integrating geometry with landmarks are actually quantitative changes, or maybe if there's something qualitative. I actually think it's quantitative, but all of these things, which tend to be parietal-based tasks, seem to be extremely impaired quantitatively, that they, even as adolescents and adults, they're at the equivalent of a 3 to 4-year-old, they're worse than their typically developing mental age matches. Whereas these things, object recognition, biological motion, language, intuitive psychology, are all as good or better than a mental age match. Okay, so what are we going to make of this? Here's a speculative hypothesis about this gene to cognition pathway. Let's imagine missing genes we know affect cellular molecular mechanisms. They have effects on the brain structure. That affects pruning, it affects connectivity, etc. But despite this, it looks like the functional development of cognitive system, even under deficit of this kind, might respect patterns of typical developmental trajectories across domains and processing streams. So I'll give you some examples. Okay, wow. This insists on making my slides skip 2 slides at a time rather than 1. So thank you for your patience. So here's an example. Suppose that in typical development, certain cognitive functions just emerge early. That would be like some aspects of language. Language is sort of almost fully formed by the time the child's 4 or 5. It could be object recognition from canonical perspectives, etc. Whereas other tasks are just late emerging. They really take a long time in typical development. So things like drawing actually have a very long trajectory. And things like the posting task, the vision for action is a little bit long, has a longer trajectory. And especially remember the non-canonical perspectives of objects. That's a very long trajectory. You don't get to adult level until you're 12 or older. So some are early emerging, some are late emerging. And this is the typical developmental profile. You have to split the tasks into early and late emerging. Now let's suppose that the gene deletion in Williams syndrome results in everything getting way slower. It's like a stretched out timeline, all right? And so we again have in black the typically developing kids with their early emerging curves for tasks that emerge early, and their later emerging curves. Now let's move that timeline over, either by moving the intercept, let's say the onset of the skill, although that's probably not the right way to view it. And we could even change the slope of the trajectory, although here I haven't. But if you just move them over and show a shallower trajectory, what you have is the early emerging things are very, very delayed in the sense they have much younger age equivalents. And the same thing happens with the later developing tasks. If you drop a line here and you say, what happens when you're 13 years old? And that's when things look like they're stabilized more or less for a person with Williams syndrome in these tasks. What you've got is an equivalence essentially to people who you're at ceiling for the early emerging things. That is to say, you're really good for canonical views. You're really good for biological motion. You're really good for certain aspects of language. And you're way worse. You're sort of at the equivalent of a 4 to 6 year old for those late developing things. So if you just draw the line here, you can see that a typically developing person who has those late emerging things might be doing as well as a person who's as well at 4 or 5 or 6 years old as a person who is 13 years old. So that's the hypothesis. It's stretched out development. It's very slow development with repercussions that the 2 sets of tasks, the early developing and the late developing, are still early and late developing, but you go over such a long time frame, and then things possibly stop, perhaps at puberty, the way was proposed that was proposed by Lenneberg for language, and there you're stuck at this level where you're in the vision for action case. You're performing, even if you're an adult, when you're posting that thing into the slot, you're performing basically the way a 4 or 5 or 6 year old typically developing individual would perform. Okay, this view makes 3 surprising predictions. I hope you'll bear with me just a few more minutes so I can show you some surprising predictions and findings. If it's true that there's very slow development followed by arrest at an early functional level, that is, you'll always be at some lower level for particular tasks, leads to surprising predictions of persistent structure, but there being quantitative limits. So first prediction is for language. The idea here is that anything that a typically developing individual acquires early should be perfectly acquired by a person with Williams syndrome. And we have evidence for that. So tense marking with regular verbs, which is acquired by 3 or 4 with typically developing kids. Every day I jump, yesterday I jumped. Kids 3 and 4 get that. And so do people with Williams syndrome. Very much more complex things like the relationships, scope relationships between terms not and or in these. And I won't go into the details of like what the structures are underlying these two sentences, but just listen and you can get different interpretations. The cat who meows will not get a fish or milk. That means he won't get either. Neither, neither, neither. Doesn't get the fish, doesn't get the milk. So or is interpreted as and. If you just move the not in the structure to a different position, you get the cat who does not meow will get a fish or milk. And that interpretation is that he'll get either the fish or he'll get the milk, not both. So very different interpretations for or depending on where the not is in the linguistic structure. That happens to be acquired by kids who are 4 and 5 years old, typically developing kids, and is also acquired by people with Williams syndrome. No problem. By contrast, things that a typically developing person acquires later in childhood, the person with Williams syndrome should never fully acquire because they're always stuck at that lower level. So examples we have in our lab are tense marking for irregular verbs. Every day I fly, yesterday the answer is flew. That's acquired somewhat late. Finalizing those representations for irregular tense verbs is a little bit later than the regulars. And it looks like people with Williams syndrome may really struggle with those. We have data now from Rennie Pasquinelli that overgeneralization of tense endings in people with Williams syndrome, which ends at about 5 in typically developing kids, persists through age 40 in people with Williams syndrome who overgeneralize. There's another example, which is different kinds of passives that are late acquired. If they're psych verbs, Bart was loved by Lisa, as opposed to action verbs, which are very easy. And there's evidence from Ken Wexler's group, Wexler and Perovich, that the psych verbs are probably never acquired in this for passives by Williams syndrome individuals. The second prediction is sensory motor mapping is really, really different. So this is just the mapping of locations in your hand, where if I tell you to close your eyes and I tap this part, and then I ask you to open your eyes and indicate to me, where did I tap? Do you get it right? We did this with typically developing kids and also adults, typically developing adults and people with Williams syndrome. And what you see up here is typically developing kids. So the accuracy for adults, a 29-year-old, it's like perfect. Most of you in there who are listening will get every single spot right. If you look at younger kids, 4-year-olds, for example, you see these really interesting errors where this might have been the target, but the person indicates this location, or this might have been the target, but this is the location they indicate. So a lot of these cross-finger errors, which have an explanation, but that's for a 4-year-old, and they resolve by the time you're in your 20s. For Williams syndrome, a 7-year-old represented here shows lots of these excursions where this finger is tapped, but the person identifies it here. This is tapped and they identify it here. This is tapped and it's identified as here. Lots of excursions and big errors. And if you look at the 31-year-old with Williams syndrome, you can see they're still making these excursions across fingers, similar to the typically developing 4-year-old. So that's really a shockingly different domain, but it's still showing this quantitative difference, that there are errors that reflect the development of a 4 to 6-year-old. And the third is a copying prediction. So I just want to show you some things you can guess at. We're predicting that individuals with Williams syndrome will have very comparable drawings to typically developing 4 or 5-year-olds, even if they're older. So you can guess, there's one set on the left and one set on the right, and you can guess which group gave the drawings, which group is the Williams syndrome group and which group is a typically developing mental age match controls. And basically, I think if you're honest, you probably won't be able to tell the difference. That's what we found with raters. But just to see if you were right, these are from typically developing 3 to 6-year-olds, and these are 6 to 14-year-olds with Williams syndrome, who look sort of indistinguishable from the much younger kids. So those 3 predictions all hold true over these vastly different domains. So revisiting and wrapping things up, the idea of having 2 different modular systems that are differentially impaired looks like it's not the right story. The empirical evidence tells us there are quite different spatial systems. We've explored a number of them today. They vary in their typically developmental timing. Some are early emerging, some are late emerging. People with Williams syndrome apparently have the right structures, the right cognitive structures to move up to the level of a, let's say, a 4 to 6 year old, observing all of the internal details of that cognitive representation, but then they don't go much further for the things that we've looked at. The data also suggests a language system whose core computational parts, creating sentences, understanding relationships between different parts of the hierarchy, constructing some aspects of tense and so forth, they also undergo slow development, and they reach the level of a typical 4 to 6 year old, but then they stop. Notice that a typical 4 to 6 year old has darn good language. Language is just about done at that point. And so they've done quite a bit. And that's why when you look at their language and listen to their language, they sound fantastic, because that's an early emerging system which has plenty of time to develop in the slow development of the Williams syndrome individual. So is it modular or is it the developmental process? I think both. And here's what I would say. All we have to do is make 2 amendments. And I'm going to say we can take both of these hypotheses for their value. The developmental hypothesis, I would say, explains the timing. It explains that the fact that when a cognitive system emerges and how long it takes to develop to full maturity is going to be affected by how prolonged your development has to be, how rapid or how slow your developmental process is. It's going to be slow with Williams syndrome, so they're not going to get quite as far. The modular hypothesis, which I'm still very fond of, I think explains a lot. It explains the structure. How did we get these structures in all of these domains that look like they reflect the same cognitive structure as for a typically developing individual? It has to be because there are aspects of these cognitive systems that just don't get changed under genetic deficit. And I would say that that fact preserves at least some of Jerry Fodor's criteria for modularity. Domain specificity, get different properties for different systems. Ontogenetic invariance, which means that things follow each other in order, whether you're under a genetic deficit or not. And robustness under deficit. They still show up regardless if you have a genetic deficit or you have a full complement of genes. So I want to just finish. Wow. Okay, by saying both of the hypotheses are important. But I want to take 1 second to give a final word, which is some implications for education, because I know that the program emphasizes education, and I think there are some important messages. I want to say that although I've been saying many of these core cognitive competencies look in adolescents and adults like they're around the level of a 4 to 6 year old typically developing individual, even though I'm saying that, I'm not saying that everything is the same as a 4 to 6 year old. They're not. They undergo puberty. They enter adolescence. They have girlfriends and boyfriends. They have social lives, etc. And they develop in many other ways, even in cognitive ways. So for example, what isn't like a 4 or 5 year old? Well, this encyclopedic knowledge and vocabulary. People with Williams syndrome, when confronted with these tasks, this is a standardized test, point to a famous building in India, very high performance on that one. So Taj Mahal. And over here, point to Victorious, very good at that one. They point to that the panel C. So they acquire vocabulary at a steady rate, and they continue to do that all throughout their lives, just like the rest of us. So that's not like a 4 or 5 year old. What's also not like a 4 year old, interesting for education, is things like reading, addition, subtraction, and other what I call tutored skills, things that you have to have interventions for. People with Williams syndrome do learn to read, many of them. It depends in part on what resources they have to draw on. Their reading is challenging for them, but they certainly get there if they have the right kinds of input and the right kinds of educational programs to train them, to teach them reading, and really are persistent at it. Addition and subtraction, they can do that. They can go up to multiplication. Many of them are in the early phases of high school math. Again, even though their approximate numerical skills are lower than you would think for their age, they still can learn some of these things, some of the addition, some of the subtraction, some multiplication, if again, given the appropriate input. And other tutored skills like map reading, they can learn to read maps. So these suggest to me that these complex skills, reading, addition, subtraction, tutored skills like map reading, for example, those depend on the foundational representations that we've talked about. And they might be assembled in lots of different ways. And they're strongly subject to formal experiences of schooling and opportunities. So that means that we shouldn't just think that people with Williams syndrome have the cognitive systems of a 4 to 6-year-old. That's not what I mean to say. What I mean to say is that some of these core cognitive systems really do look like that in terms of quantitative change, and that we need extra intervention in order to bring them further along in other things that basically all typically developing individuals need input on and tutoring on and formal education for. So with that, I'll say thank you to all of you, and thank you to my many, many collaborators who have been in this with me for many years. Jim Hoffman up at the upper left sort of started this out with me. Katrina Ferrara, Kristen O'Hearn, there's Danny Dilk, some of you may know, Julian Mussolino, Laura LaCosta, Rennie Pasquinelli, Freddie Camps, and many, many other people. It was a huge effort, funded by a variety of people. Thank you so much, and apologies for the crazy cursor, which I'm going to have to rewrite. And I'd be happy to take questions if there's still time.
Thank you so much, Dr. Landau. Fascinating presentation. And I think I could, watch this all day. So interesting to be able to learn all of this information for you. So again, thank you so much for this presentation. If you could now close out your slides,
Sure.
Stop sharing your screen, that would make it easier for all of us to see with the Q&A session. And just a reminder, if everyone has questions, we do have a few minutes remaining. We have interpreters for roughly 10 more minutes. So please, if you do, post your questions in the Q&A.
And by the way, if anybody doesn't get to ask a question they'd like to, they should feel free to write to me. I know this took a lot longer than it should have, so I apologize for that, but yeah.
Completely understand. So the first question, just a moment here, is talking about if there have been any studies with deaf people who use ASL, because it's a visual spatial language. And I do want to add a little bit to this as well. I'm wondering about if there are conditions where, for example, you were looking at the biological motion perception with the point lights, and you had 3 separate conditions. You had conditions 1 through 3 there with the yoked condition, which was the most difficult. I'm thinking about that, and I know that deaf people would generally show better performance than other typically developing adults.
Yeah, I think that's a fascinating question, and to just go up to where you began, and to think back probably 30, 40 years when Belugi started this work, she actually was incredibly interested in seeing whether she could find somebody who had Williams syndrome who was also congenitally deaf, for exactly the reasons that you're thinking about, and exactly the reasons, of course, that have occurred to me as well. I think it'd be a fascinating question because signing is language, and yet it's spatial. And I can tell you that my strong prediction, based on all the work that we've done, would be that sign languages if you have Williams syndrome and you're congenitally deaf and you have exposure to sign language, your language will develop at the same rate as a hearing speaking child in a hearing speaking environment, and that there shouldn't be any particular points of problems, the only possibility might be if there are things that depend on mirror image reflection, which is the very, very hardest thing for representation, no matter what. If there's some marking that depends on that kind of mirror reversal image, there might be some implications. But I would say that even for very complex structures, they should be totally fine, because language, I think, is a modular system. It's got its own properties, and whether it draws on a visual manual mode or an auditory speech mode, it should be the same. I do think, by the way, as I'm talking about this, there was a person in Britain who might have found, I think it was Betsy Wall, might have found a person with Williams syndrome who was a signer. However, the problem is that at the time that she was, that this came to my attention, I don't think, there are different ways of diagnosing. You have to be really careful about diagnosis and be sure that it's a person who really has Williams syndrome. So you have to do the right genetic tests. And I think there was some question about whether this person actually had Williams syndrome. So that's number one. The second thing about biological motion is a fascinating question, and I agree with you. I have a collaboration, the collaboration on perinatal stroke is with Lisa Newport and her group at Georgetown. And there's a student there who's actually very interested in, we're developing more biological motion tasks to be doing brain imaging on both perinatal strokes of the right or the left hemisphere, but also on young, typically developing healthy kids. And this student is actually very interested in pursuing the question of how do these very, very complex cases shake out with a person who is a deaf native signer. So I think it's a great, those are fantastic questions. And, if anybody's ever interested in trying to find an individual who has Williams syndrome, who's a deaf native signer, and if you do find such a person or want ideas about finding and want to collaborate, I would love to do that. I think it's a great question.
I don't know of any specific individual with Williams syndrome, but in our program, we do have faculty who have studied biological motion specifically. Dr. Lorna Quant focuses on that and does EEG studies with that. And she has a recent publication as well that demonstrates with deaf people specifically how they process biological motion better than non-signers do.
Fantastic. Do you know, well, I can look her up. That's great. I'm not familiar with that paper. The student might be, but I will look that up for sure. Thank you.
And Dr. Quant is here, Q-U-A-N-D-T.
Right, I have that written down. Yeah, thank you.
Right, let me check and see if there are other questions. There's a longer question here, so I may have you read that if you wouldn't mind, from Kara.
Where is it?
In the Q&A.
I see in the Q&A. Oh, I see in the chat. Sorry, I was looking at the chat.
In the Q&A,
okay.
yeah. No,
I see Kara Hawthorne. Okay, I'm curious what pattern of results absent structure. Yeah, right. Recognizing objects, participants. Right. Good. Thank you for that question. So Kara's asking, what evidence would we want to say like it's absent structure? So let me deal with that first, and then I'll tell you about what we have that you want for the object recognition task. The thing that really set us back on our heels was when we thought we discovered that people with Williams syndrome simply couldn't compute geometry at all. That was in the case of the very first study that we did in 2010 before we sort of tweaked things. And that would be a case of if we had pursued that and continued to find they do not represent geometric structure, that would be absent structure. So that's a strong example. I mean, another example is if you could never do the biological motion task, you could never extract the point light walker and determine which direction it's walking. That would be a case of absent structure. In the case of the object identification, we actually did look at types of errors and correlation between which items were easy and which items were hard. And there was a high degree of correlation between the people with Williams syndrome who made errors and the typically developing controls, all the groups. So people, everybody found some of them really hard and some of them really easy. And there was a high correlation there. And the errors that they made, or the actual responses. So the task is actually a naming task. So, you have to say cauliflower, but we would also take vegetable as an answer. So you didn't have to say cauliflower. Or if we said, I don't know, actually in the first object that we saw, I said violin, but I don't really know, was it a viola? So, so somebody, if we showed a bass and they said violin, we would take that. We would even code, but we would code if they said musical instrument. So we didn't give them full credit for that. So we also analyzed the types of errors, the types of responses that happened, and they were actually really comparable. There weren't any differences between the answers that people were providing, the names they were providing, and other groups' answers. So that suggests to us that they really are recognizing the object. And of course, there are many other things that we could be doing to be asking, are they really as good at every single orientation change? We don't know for sure. They're probably worse at left at mirror image. Actually, we have other data that suggests for non-real objects, for made-up objects, simple geometric objects, they're very quite bad at mirror image reversals. So there are some differences, but I think as far as we've gotten, we very carefully checked to see that it wasn't just that they were doing it, it's that they were really doing it with, as much as we could tell, the same kinds of processes that are used to recover the shape. Remember, in the non-canonical, you have to also recover the shape. It's not as if they're doing nothing in that task, by the way. So chance level is minuscule because you have to name it and get it right. So so even in those cases, they dropped, but they were down around maybe 50 or 60%. Yeah. So I hope that helps.
Yes, that was wonderful. Thank you. I had the same line of questions. I think one final question, and then we will close our webinar today. The last question is from an educator who was wondering about interventions, and as an early interventionist is asking what type of things should we be doing to help children with Williams syndrome in developing these skills that you've noted. What suggestions do you have?
Thank you for that question. I think it's so incredibly important. I can think of two classes of things. So of course, it depends on what skill or cognitive function you're trying to improve. But let's just take math. We know from the kids that we've looked at that they seem to be doing some of the formal math problems like addition, subtraction, using symbolic somewhat differently. They may rely more on language than their approximate number system. So remember, no reason for you to remember, I'll remind you, in the approximate number task, we actually found that adolescents have a Weber fraction or a sensitivity to numerical approximation that's about equivalent to a 6-year-old. 6-year-olds are not doing multiplication, but people who are adolescents with Williams syndrome can do multiplication. So something's going on there where they have to use something about the approximate number system, maybe, but it's not clear what. So people like Stan DeHaan, and also I think there's work from some of my colleagues, Justin Halberta and Lisa Feigenson at Hopkins, that look to see the kinds of training programs you can use to improve mathematical skills by, for example, by improving your approximate number sensitivity. And the extent to which that generalizes to formal math is still unclear. But I think that line, and I would look at Stan DeHaan's work in particular as an option, because he really is very serious about interventions for math. The other thing, I'm going to give two other examples just quickly. A second example is reading. And we found consistently, and this is only on an observational level, but it really resonates. Some of the individuals that we've seen over the years have had very active interventionist-minded parents, and they find one of the schools that was touted was Lindemood Bell. I don't know how effective that is in general, but this particular family found it enormously effective. And that individual who we studied for her reading really could do a pretty good job of reading. She was, I think, at 3rd grade level when she was 15, which is pretty good for this population. And so I would actively investigate, any of the standard interventionist therapies that people think matter. And keep in mind that for an individual with Williams syndrome, it's probably not going to be the phonological part. It's probably going to be the visual spatial part that's a problem. So if that's true, there should be exercises that can help you distinguish between the B's and the P's, et cetera. That would be a second thing. And then the third thing I just have to bring up, because it's something I worked on for a while, and then we dropped because the engineer I was working with left to go to Stanford. But this was a project of my heart. We wanted to see whether we could help people with Williams syndrome just draw better straight lines. And this was on the way to drawing better triangles or drawing better circles, whatever. And what we did was, this engineer was developing force field mechanisms where you would put a pen into a little stylus holder, and then force fields, the person would go to draw a line, and force fields would force them back and forth so that they maintained a straight line. And we did that with, we had a poster on this. I think there were 4 or 5 participants. And it seemed that, at least in the first trials that we were doing, that actually had some impact on getting them to have control over their pencils, to know the visual spatial, routine that you use when you're drawing a straight line. And then the question would be, can you use something like that? So if you had engineers around you, I would go find them, because they're really good at this kind of thing, and think about, for the visual spatial kinds of things, ways of intervening in that way. So thank you for the question again. I think the interventions are so important, and we always hope that what we're doing is at least somewhat, provides at least somewhat useful insights. Thank you.
Great, thank you so much again, Dr. Landau. And I think our time is up for today, unfortunately. I'd like to thank our interpreters as well. I always forget to thank our wonderful interpreters, so thank you so much to them. And thank you to everybody who joined us today. And if you have any further questions, feel free to email me, and I'm happy to forward those questions on to Dr. Landau, or connect you with her if you'd like more information. So thank you again, Dr. Landau.
Thank you so much.
And
I really appreciate it. I wish I could be there in person.
Right, I hope that you can someday. I hope so as well. Hopefully, yes, it would be great. I look forward to that.
Good. Thank you so much to everybody, and thank you again for your patience.
Oh, absolutely. And thank you so much. We'll go ahead and